So now we are going to study the process of translation. Firstly, I'll describe the different steps involved in the process of translation, specifically in the prokaryotes. The first step is the charging of the tRNA. So as we know, tRNA binds with the amino acid, and with the presence of ATP molecule, one ATP is involved in this reaction, it becomes amino acyl tRNA and it releases AMP. The most important thing is this amino acyl tRNA is unique for each amino acid, because there are 20 standard amino acids are used, right? And these are synthesized by the total 64 codons, out of which 61 codon code for amino acid and 3 are the stop codons, right? And 1 start codon, AUG, which codes for the methionine, and it is included in the 61 codons which code for the amino acids. And the specificity of tRNA depends on the either size, charge and R group of the amino acids. As we know that amino acid have different groups, R group is there, then amino group is there, carboxyl group is there, hydrogen. So this is the basic structure of the amino acid, right? So the type of R group involved in the amino acid affects the specificity of the tRNA. Charges, number of charges present on the tRNA, whether amino acids are negatively charged or positively charged, then again the size of the amino acids, right? Now coming to the involvement of ribosomes. Ribosomes are the important part which are involved in the process of translation. So first of all, you must have seen the structure of ribosome, right? It is having a smaller subunit and larger subunits when we talk about the prokaryotes as well as in eukaryotes. So structure is like this, which is given in most of the books, and if you can see, this is the smaller subunit and this one is the larger subunit, right? So overall you must have seen that it is 70s in case of prokaryotes and 80s in case of eukaryotes. So we are talking about here prokaryotes, so we will be talking about only the 70s. As you know, this is Swedberg unit, inspired from the name of the scientist who was working with the ribosomes. So we will not go into the detail right now. So what happens if we dig into the detail of this structure of ribosome? So one by one, when I'll be explaining the steps, I'll show you that how the smaller subunit and larger subunit of the ribosomes are involved in this process of the translation, right? So coming to the initiation steps, first of all, important thing is what are the components which are required in the process of the initiation of translation. So very important step, very important component, that is the mRNA. mRNA is the most important component which is required for the process of the translation, right? Then again, which we have discussed, the smaller and the larger subunit of the ribosome, and again one of the important factor, that is the initiation factor. So there are 3 initiation factors which are used in the process of translation in case of prokaryotes. Initiation factor 1, 2 and 3. So we will be discussing one by one these initiation factors, right? Apart from that, we do require initiator tRNA with N-formyl methionine attached. So this is the charged tRNA, right? It has important role when it attaches to the tRNA, attaches to the smaller subunit at one particular site, which we will discuss later in this lecture, right? Then again, we do require GTP, right, for the energy. component in this step for initiation, right? Then comes the initiation factor, which is very important part. We have discussed the initiator, we have discussed the GTP. When we talk about the initiation factors, what are their roles, how they are impacting the initiation of translation in prokaryotes. So first of all, initiation factor 1. Initiation factor 1 enhances the process of dissociation of the large and smaller subunits, right? So it confirms, or what it does, it separates, right? It ensures that smaller and larger subunit of the ribosomes are dissociated from each other, right? Then comes the initiation factor 2. It binds to GTP, right? And it helps initiator tRNA or formyl methionine to attach with the initiation codon, which is AUG in this case, right? And it codes for the methionine. Clear? This I have told you earlier in this lecture. And again, then comes the initiation factor 3. It binds to the smaller subunit of ribosome and it prevents the large subunit from binding during the initiation. Clear? So you must not get confused when we study the function of initiation factor 1 and 3, because 1 is involved in the dissociation, right? And the 3 prevents the larger subunit from binding to the initiation. It binds to the both of the these initiation factors 3 and 1 bind to the smaller subunit of the ribosome during the translation process, right? Then coming to the elongation, again we will be discussing about the components which are required in the process of elongation. So 70X S complex, ribosome complex, tRNA charged with amino acid, then elongation factor. In case of initiation, we do require initiation factors, but over here we require elongation factors, then GTP for energy source, right? As I have told you, there are certain sites which are involved in the process of elongation of translation, right? And these are the amino acid site, peptidyl site, exit site. Now the question is where these sites are present. So I'll try to make you understand that where these sites are present, right? So these are the ribosomal subunits. You have seen this is the smaller subunit, right? This is the larger subunit. Okay, so we can see this is the mRNA, right? mRNA is binding to the smaller subunit, and if we talk about the initiation codon, right, this is AUG. This is present somewhere here, initiation codon, right? So these are the 3 sites which are present. A site is present here, right? Then comes the P site. P site corresponds to the initiation codon, and E is the exit site, right? This is exit site, this is peptidyl site, and this is the amino acid site, right? So the charged tRNA first binds to the A site, right? Then it recognizes the initiation codon and it starts the process of translation, right? When translation happens, after the last step, it gets removed the polypeptide sequence from the exit site. tRNA removes from the exit site and polypeptide chain is removed, right? Clear? Okay, so if we go into the detail of process of elongation, right, so we can divide this into the 3 steps. The one is the charged tRNA enters into the A site, which I have discussed, right? Over here on right hand side, amino acid site, charged tRNA enters at this site, right? A peptide bond is created between amino acids in the A and P site, right? Over here the peptide bond is formed, right? Now the question is, what is peptide bond? So I can tell you that this is the bond between the adjacent amino acids in the polypeptide chain, right? So how this bond is formed, you have to remember that polypeptide bond is always formed. Let's say this is one amino acid, right? As I have told you earlier structure, this is carboxyl group, this is amino group, right? This is the amino acid 1, right? And this is the amino acid 2. Any amino acid could be there, right? So this bond is formed between these two components, right? Over here, this bond is known as the peptide bond. Clear? So it is formed between the carboxyl end of one amino acid and amine group of the second amino acid, right? So this is what peptide bond is, right? Now coming to the third point, we have already discussed first and second. Now the third point is ribosome translocate to the next codon, right? So these are the three steps which are along in the elongation step, right? Now coming to the three elongation factors which are involved in this process, right? So elongation factor TU, right? A charged tRNA binds to the A site, right? This binding takes place when elongation factor TU joins with the GTP, right? It is the source of energy, and then with the charged tRNA to form the three-part complex, right? So if question is the elongation factor which is involved in the formation of three-part complex, it is the EFTU, or which elongation factor required the GTP as a source of energy. So this is again elongation factor TU. Clear? Either 1 mark question in MCQs. This always examiner asks these questions, right? Then the elongation factor TS, it regenerates EFTU GTP to EF GTP, because when energy is consumed, then GTP converts into GTP, and this forms a complex with elongation factor TU, right? Then again what happens, the second elongation factor, which is elongation factor TS, converts again it into the EFTU and GTP. This is the again active form, right? It can again take part in the process of elongation, right? Then comes third elongation factor, G. So it helps to position the ribosome over next codon. So there is typo error, right? Over next codon. So basically it helps in the process of translocation, which you have to remember, right? It helps in the process of translocation. Clear? So now we will move towards the next step, which is the process of termination. So what happens? Termination takes place when ribosome reaches to the termination codon. So as I have mentioned earlier, I have told you the initiation codons. So the termination codon, there are basically three termination codons which do not code for the amino acid. So three codons which are used or considered as termination codons are UAA, UAG, UGA, right? And other name for this amino acid is ochre, burr, and opal, right? Sometimes this question is asked for 1 mark in either MCQs or one liner, that what is ochre, amber, or opal. So you have to tell the sequence. This is the UAA, UAG, and UGA. You have to remember that, right? So what happens? Release factors bind to the termination codon and causing the release of polypeptide from the last tRNA of this tRNA from the ribosome and of the mRNA from the ribosome. Clear? So this is how termination of translation process in prokaryotes takes place. So when we talk about the components required, so termination codons we require, which I have discussed here, right? Then the release factor required. These release factors are, firstly, three release factors are used. So RF1, it binds to two codons. The first one right? RF1 binds here. When we talk about RF2, it binds here and here. So release factor 1 and 2 bind to the UAG or amber, right? These codons, clear? And RF1 only bind to the UAA, RF2 only bind to the UGA, clear? Then comes through the release factor 3. It binds to the ribosome and forms a complex with GDP, and this is how it releases the RF1 and RF2 from the complex, clear? An important thing is RF1 and RF2 binds to a site of the ribosome, which promotes the cleavage of the tRNA in the P site, right, which I have told you earlier, from the polypeptide chain, and the release of the polypeptide happens over there, right? So this was all about the process of translation in case of prokaryotes. And if you have any query regarding this lecture or any question, you can write down comments. And that's all for the today. Thank you very much.